My name is Jessica Wehinger,
I'm Dominic Schuh.
And I'm Ethan Daniel.
and we will be presenting on the distributions and frequencies of pathogenic gene mutations for Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease, the first being Alzheimer's disease, and it affects approximately 1% of people over the age of 60. Unlike Alzheimer's disease, Parkinson's is a motor disease that is clinically characterized by bradykinesia, rigidity, tremor, and postural instability. It is known that these symptoms are due to the death of dopaminergic neurons in the substantia nigra region of the midbrain. However, it is not fully understood what causes these neurons to die. Furthermore, as many as 70% of dopaminergic neurons can die before clinical symptoms present. Studies have found mutations in several genes that are either causative or serve as risk factors for developing Parkinson's disease. The aim of this project is to study the global distributions of these disease-associated mutations in order to see which populations, if any, are at a greater risk of developing Parkinson's disease.
So here's our first gene, SNCA. This gene codes the protein alpha-synuclein. Alpha-synuclein is thought to help to regulate the release of dopamine in the neuron synapse. Mutations in the SNCA gene are autosomal dominant, meaning they result in a gain-of-function mutation. This unwanted gain causes the alpha-synuclein to aggregate into balls of phosphorylated protein called Lewy bodies. Lewy bodies are the hallmark of a Parkinson's diagnostic that is confirmed post-mortem. This is the table summarizing the frequencies of SNCA mutations in Parkinson's patients around the world. Most studies found no mutations within the SNCA gene. For the American and Greek study, participants were either Greek or American with Greek heritage. Many studies did not have this type of data though, and those countries are seen here in the light gray. You can see most cases that do have the SNCA mutations are from somewhere in Europe, but even so, these frequencies are low.
The second mutation that we studied was LRRK2, which is leucine-rich repeat kinase 2, which is a gene that encodes a protein called dartarin. Dartarin is a kinase that phosphorylates proteins involved in autophagy and vesicle trafficking, including alpha-synuclein. LRRK2 mutations are some of the most common mutations found in Parkinson's patients globally. And of the LRRK2 mutations, G2019S is the most common mutation found in populations of Parkinson's patients. The G2019S mutation has been found in approximately 20% of Ashkenazi Jews with Parkinson's and in approximately 40% of North African Arabs with Parkinson's. Review of the literature resulted in the following distributions.
PINK1 and Parkin are genes that code for proteins that function in the same metabolic pathway. In a healthy cell, PINK1 codes for a mitochondrial kinase. This kinase phosphorylates the Parkin protein, which then helps ubiquitin flag damaged or dysfunctional mitochondria. So in cells that have mutated PINK1 or Parkin, dysfunctional mitochondria are allowed to build up, and the cell or neurons with Parkinson's can't produce enough energy. As this is a loss of function mutation, the mode of transfer is autosomal recessive. Here the table summarizes national PINK1 and Parkin mutation frequencies. The first 6 studies looked at the entire gene, while the remaining studies looked at a single mutation within the gene that is commonly thought to cause Parkinson's. But however, due to these specific studies that haven't found many instances of the gene, the role of this mutation in causing Parkinson's is under question. This map shows the studies that looked at the entirety of the gene. And another point to add is that Park1 mutations typically have a younger age of onset than just in normal Parkinson's disease.
CHCHD2 codes for a mitochondrial protein of the same name that has many mitochondrial regulatory roles. These roles include maintenance of mitochondrial crystal structure, regulation of electron transport, suppression of mitochondrial-induced apoptosis, response to oxidative stress, and cellular migration. Mutations in CHCHD2 have been shown to result in mitochondrial dysfunction, which in turn affects energy production. Dopaminergic neurons are very energy intensive, and their death could be a result of impaired energy production by dysfunctional mitochondria. Mutations in CHCHD2 have been found primarily in Asian populations, specifically Japanese populations, and are absent in many other populations. The following map shows the global distribution.
The final enzyme that was studied was GBA. GBA codes for an enzyme called glucocerebrosidase, which is active in lysosomes, the cellular compartments that are responsible for digesting old cellular components, pathogens that enter the cell, and toxic substances. Glucocerebrosidase breaks down glucocerebroside, which is a component of the cell membrane. Mutations in GBA cause Gaucher's disease, which is a buildup of fatty substances in organs, including the liver and the spleen. Though not directly implicated with Parkinson's, like some other gene mutations in GBA do increase the risk of developing Parkinson's. Those with Gaucher disease and those who are carriers of GBA mutations are more likely to develop Parkinson's. As you can see from this table that summarizes many countries around the world with frequencies of GBA mutations in positive PD cases versus those in control cases. The two most commonly linked mutations were the L44P mutation and the N370S mutation that appear to be linked to Parkinson's occurrences around the world. This map displays several of the countries listed on the table. And as you can see, there are several that lie in the 2 to 4% range that summarizes a relatively high statistical correlation between these mutations in GBA and PD occurrences.
As stated earlier, much is still unknown about the exact causes of Parkinson's disease. Populations around the world are aging, so it is expected to see a rise in the number of Parkinson's patients in the coming decades. While there are treatments for Parkinson's, there is no cure, which, coupled with the expected increase, makes research into the field crucial. Genes only indicate a heightened risk for developing Parkinson's, so knowledge of environmental influencers will be necessary to understand the full picture. Though not definitive indicators for the development of Parkinson's, understanding how and where disease-associated mutations occur will help to elucidate the pathogenic mechanism for Parkinson's. This knowledge can in turn be used to develop new therapeutics, diagnostic techniques, and eventually a cure.